This summer, we were able to present a revised classification of AML genetics that was really inspired only through unsupervised learning technology. This year in the ASH conference, we were able to validate the relevance and the mapping of this classification on a multinational diverse cohort. And we think that this is supporting the evidence that we generated here in the classification and in the understanding of AML biology. We have several classification systems currently for decision-making, which are especially risk classification system. In particular, and this is really clear to name, the ELN, the European Leukemia Net algorithm of 2012. It's very popular and it's also very efficient because it utilizes just 3 risk categories of favorable risk, intermediate risk, and adverse risk. And this allows the stratification for patients going into consolidating treatments with stem cell transplantation and those having also a very high risk of relapse, and for people who will be just receiving chemotherapy without further cellular therapy. Now, this system works very well for a lot of patients, but we were able to showcase that refinements in addition to the system could be provided with our system too. One needs to keep in mind here that treatment response and the biological classification do not always go hand in hand, because treatment response is changing with the therapeutic landscape. Today, for example, we have very specific inhibitors available for certain subgroups. We know already that FLT3 mutated AML can be inhibited and IDH mutated AML can be also targeted, but we have now for KMT2A and NPM1 mutated AML also menin inhibitors available. So this is a current very much changing therapeutic landscape, although not all of the inhibitors are currently approved for the routine care and still under investigation. The Harmony platform is a very important and publicly available tool. It's contributing to the advancement of research and clinical care in hematology. I think one of the big advantages, utilizing especially the machine learning predictors that we have available on the Harmony platform, is that you can map your patients and your patient outcomes more specifically and targeted to their genetic constellation in terms of mutation, beyond what we have currently available for example, the risk classification according to the ELN risk. In particular, you mentioned that it is a diverse and comprehensive database. This is exactly true. It's not only data from Germany or data from France that went into this model, but it's really an international cohort, first of all on the European level, but also beyond. And I think this is something that clinicians worldwide can build on. I think you are referring now back to the Dirichlet or unsupervised learning classification that I presented at the first step of our interview. We utilized a very broad approach, integrated 55 different gene mutations, and in addition derived 31 categories from the cytogenetic itself. So cytogenetics was really leveraged here because it's a rich source of genetic information also in AML, and also a very, very diverse source of genetic information, because patients present with all kinds of abnormalities. We think that this very broad data set and this broad use that covers even rarer variants is really an advantage, because it comprehends better to the complexity and broadness of the heterogeneous disease, which is AML. I'm really happy that you're asking those two questions, because indeed those are important. The one is important for data science because dealing with missing or heterogeneous data needs to be done in a careful way. Indeed, we had a small proportion also of missing data in the Harmony derivation cohort, and we were very careful utilizing very established data imputation approaches from the data science, meaning that certain informations in the genetic terms were indeed added from the distribution that we found within this cohort and also other established cohorts. But this was only done for a very small proportion of data, beyond 1% per class. So it's a very, very tiny adjustment. I think that the very broad and integrative genomic data that we have available, and also the care for the complexity and heterogeneity of the subgroups, not only from the type of its genetic information, but also with the origin of the subgroups, meaning in terms of countries and ethnicities, will really improve the understanding of AML genetics across continents. And this is also what we've seen in our validation cohorts, where we were able to integrate patients from East Asia, but also from America and also from different European cohorts. So we had a very, very broad data set here. I think this is an important question that you're asking, but we are prudent to answer it at this specific point. One thing that we have seen is that we are able to subdivide certain classes that are well established. For instance, the classes of NPM1 mutated AML, but also for patients carrying the inversion 16 AML, and also for those with myelodysplasia-related gene abnormalities. For these three groups, we were able to subdivide them further. And what we hope to be able to show with our final analysis is that those differences also translate into differences in outcome. We have seen this already for some subgroups and are still working on confirming this broadly. So I keep it at this today. Is my statement still a bit prudent? But I think the insights into the biology are already very important. You named it, because this is one of the most important clinical results out of such works. When we define more clearly and more precisely, because this is what it's all about, it's precision medicine inspired by big data. When we are able to guide our patients more precisely to certain treatments, we are also able to design clinical trials that are specifically targeting those subgroups. And particularly, this could be true for subtypes of NPM1 mutated patients on the one hand, but also for other patients that we have been identifying. It could be really interesting to see whether the affinities of existing drugs are also different in those subgroups. So there's still a lot of work for the clinical development, but also for the preclinical review of those subtypes that can be very meaningful and advanced drug development indeed. I think we are already in the middle of it, at least in the high-income countries. This is a lived reality. We can say that since the Human Genome Project or the TCGA project, which is including it, the Cancer Genome Atlas, just to name it for those who are not familiar with it, this is really a starting point over 10 years ago that the large and multi-gene data was implemented into medicine. And for AML, this is a journey that is continuing, and I think it is getting an additional velocity now with our new results. So we are already in the line. I'm really a very great fan of Harmony, and not because it's a large and European research collaborative, but because it's so incredibly inclusive. Research proposals are provided by very experienced and established researchers in the field, but also by younger postgraduates or graduate students. And those research proposals are driven by the possibilities that are attached to data science and to the prospects in precision medicine, but also they are accompanied by senior experts in the field and continuously reshaped. What I want to say is that the research environment is very rich and it's healthy. And it is committed to prospect and advancements in care, ultimately to the patients and to their well-being.